Hello, I am Dr. Stephen Newmaster. I'm professor of genomics at the University of Guelph, and I've spent my career creating molecular diagnostic tests for different types of species, from plants to viruses. Today, I'm going to talk to you about how do we actually identify the corona 2 virus. And I have to start by talking a little bit about virus biology. The corona 2 virus is really made up, it's quite a simple structure with a complex function. And it's made up of a circular protein coat with a genomic RNA strand inside. So, as a molecular diagnostic scientist, there are a number of different ways to be able to identify this organism. The first way is go after the genome. You break open that capsule and you can identify the RNA. And we use this in a polymerase chain reaction test. We have segments of the DNA, which are primers and probes, and they identify that specific virus. And that test, that PCR test that people are familiar with, is very, very sensitive. There are different types of assays that we design, so not all tests are created equally, and the most sensitive ones can actually pick up asymptomatic patients. When that virus first enters your body, it starts to populate your body and it can be picked up by the PCR. The second way to do it is that protein code that I talked about. An antigen test has certain structures that it picks up on the protein code around the virus. And when your body has enough of the antigen, enough of the virus in the body, then you can do this test. It's a shorter amount of time. It's a very accurate test, and it's quite sensitive, and it's very specific to the strain of the virus. But it has a very limited amount of time that test is effective. Lastly, the response of your body, not identifying the actual virus in your body, but the response is the antibodies. And your body produces a number of different antibodies over time after the virus has actually infected you. The first ones are the IgMs. And that is a short-term response. The body goes and attacks the virus and breaks it down. And the second antibodies that come in, the IgGs, are the long-term response. And they actually give your body some immunity. That test happens in one shot normally, those best tests. And not all antibody tests are created equally. Some are more sensitive. Some are more adapted to different virus strains. The antibody test tells the patient that you have had COVID at one point in time and you have some resistance to it based on your IgG levels. These are important tests to think about in timing and when we would use them to diagnose early nascent foci or early populations, and this is critical for controlling outbreaks.